Good morning, class. Today's topic is the Human Genome Project. Human Genome Project. Genetic makeup of an organism on an individual lies in the DNA sequences, and all genes together of haploid set of chromosomes are known as genome. So this is the definition of genome. The differences in two individuals will naturally be reflected in the differences of their nucleotide sequences. This can be known only if the entire human genome is mapped. With the establishment of genetic engineering techniques, where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining the DNA sequences, a very ambitious project of sequencing human genome. It was launched in the year 1990. US government started this human genome project, and it is coordinated by the Department of Energy and the National Institute of Health. The goal was to complete the mapping and understanding of all the genes of human being. So Human Genome Project was a 13-year project coordinated by US Department of Energy and National Institute of Health, and it is a mega project. It is known as a mega project. Reason behind this that human genome have approximately 3 into 10 to power 9 base pair. The cost of sequencing required near about $3 per base pair. Then if we are going to estimate, the total cost of this project is 9 million US dollar. The obtained sequences were to be stored in a typed form of book. If we are going to type it in form of book, and if each page of the book contains 1000 letters, then each book contains 1000 pages, and the 3300 such books need to be stored information. So you can imagine it is how it is known as a mega project to form a single human cell. What are the aims and goal of this project? First aim is to identify the approximate 20,000 to 25,000 genes present in the human DNA. To determine the sequences of 3 billion bases that make up human DNA. And third aim, to store this information in the databases. And next is to improve the tools for the data analysis. The last and the very main is to address the ethical, legal and social issues that arises from the genome researches. What methodologies are used for this human genome project? The method involved in the two major approaches. There are the two major approaches. One is the identifying all the genes that expressed as RNA. We can say it is a expressed sequence tags, ESTS. This is the first technique. And the second approach is to blind approach of simply sequencing the whole genome that contain all the coding and non-coding sequences, later assigning different regions in the sequences with the functions, and this is known as the sequence annotation. So two methods, sequence annotations and the expressed sequence tags, two methodology is used for this project. What are the salient feature of human genome? Basically, the human genome contains 3,164.7 million nucleotides bases, and the average gene consists of 300,000 bases, but the sizes are varies. Among all these, the largest known human gene being dystrophin, and the total number of genes is estimated near about the 30,000. It is lower than the previous estimated. Previous estimated was the 80,000 to 1,40,000 near about genes, but the total number of genes is estimated by this project is 30,000. Almost all, means 99.9% nucleotide bases are exact the same in all peoples, and the functions are unknown for over 50% of the discovered genes. 50% genes functions are yet to be discovered. It is unknown, and less than 2% of the genome code for the protein. As we know that chromosome number 1 have 246 million base pair. So it is the largest genome in the chromosome number 1, and the lowest is only 50 million base pair is present in the Y chromosome. Short stature Y chromosomes, Y-linked short stature chromosome. Repetitive sequence make up very large portions in the human being. So repetitive DNA is too much. There are too much repetitive DNA sequences are present in the human genome. And chromosome number 1 has the most gene, too much genes, near about 2,968 genes are present on the chromosome number 1, and the lowest is 231 genes are present on the Y chromosomes in the human genome. Scientists have identified about 1.4 million locations where the single base DNA sequences are present. Single base DNA sequences are present about 1.4 million locations in the human genome. It is known as the SNPs or SNIPS, SNPs, single nucleotide polymorphism. It is known as the single nucleotide polymorphism used in the biotechnology. In the field of biotechnology, it is very, very useful. This information help to finding the actually chromosomal locations for disease associated sequences. If we are going to trace the disease associated sequences related to the genes in the human history, so this information is very, very helpful to finding the chromosomal locations for the disease sequences, associated sequences. Before wind up this topic, there are few applications of this project and as well as some future challenges also. In applications, we know that the completions of first phase of human genome project has been compared to discovery of antibiotics, because it has opened a vast database of knowledge about the various aspects of the human genome. There are more than 1200 genes that causes the common cardiovascular ailments, endocrine diseases like diabetes, Alzheimer, cancer and other neurological ailments that can be corrected with the help of this project. With the discovery of this project, it will be possible to study the interactions between the various genes. So with the help of this result of this project, we will study the interactions between the various genes, proteins, as well as the mechanism of forming the tissues, organs, etc., etc. It also holds the promises of healthier and longer living. This is the future challenges. The future challenges for this project, that it holds the promises of healthier and longer living, designed drugs and genetically modified diets according to need of the individual human being. So these are the future challenges. Let's see what happened to the next. So this is all about the Human Genome Project. Thank you and have a nice day.